Hi guys, my name is Nika and I am one of your SIs for this quarter, and today we're going to be going over cellular respiration. So I created this chart for you guys. I think this will be really helpful. It pretty much separates each stage of cellular respiration and requires you to write the products and reactants of each step and then the location for them. So I definitely recommend that you guys fill this out and use it for your midterm, since you have everything on a sheet of paper and it's also easier to study this way. So as Dr. Joseph mentioned, cellular respiration occurs in 4 phases. And with each of these stages, they occur in different areas within the cell, and they also have different reactants going into them, right? But if you were to add up all of these individual reactions, you would still get that the same cellular respiration equation I'm sure you guys have seen before, which is C6H12O6, which is glucose, plus 6O2, which generates 6CO2, 6H2O, and ATP. So I kind of wrote down each of these steps beforehand because it would take me a while to kind of write this with you guys. But let's go over glycolysis first, right? So glycolysis occurs in the cytosol of cells, and we take glucose and we break glucose down into 2 pyruvates. Remember that since glucose is a 6-carbon molecule, we're going to break it down into 2 3-carbon molecules, which is known as pyruvate. And when this happens, we generate ATP and NADH, which we will talk about later in this video. So you should have this in your chart. So your glucose, which is 6 carbons, makes 2 ATP, 2 NADH, and this is all happening in the cytosol. So now we have pyruvate oxidation. So now we're going to take our pyruvate, which is in our cytosol, and do something to it. So we take the pyruvate and we make acetyl-CoA, NADH, and CO2. So as you guys can see in the reactant side, we took the pyruvate, which is a 3-carbon molecule, and with the help of an enzyme called coenzyme A, we're going to take this 3-carbon molecule and make it acetyl-CoA, which is 2 carbons. So because of this reduction or loss of carbons, we're going to generate a CO2. And also through this process, we're going to make NADH. Location-wise, remember that the pyruvate was in the cytosol, and now it's going into the matrix of the mitochondria, right? So kind of wrote it in the second column. We took the 2 pyruvates. We don't make ATP, but we make NADH, acetyl-CoA, CO2, and this is all occurring in the mitochondria. So notice how my mitochondria is in red. From now on, all the other steps, so citric acid cycle and oxidative phosphorylation, will be in red because it occurs in the mitochondria. And remember that if it's in the mitochondria, it requires oxygen, right? So now I'm talking about a cell that has oxygen. So the next step is citric acid cycle, and this is when we take that acetyl-CoA that's in the matrix and we break it down into carbon dioxide, and in this process we make NADH, FADH2, and ATP. So if we go back to the table, I wrote acetyl-CoA is the reactant. We make 2 ATP, 6 NADH, and 2 FADH2. And in this process, we're breaking it down into carbon dioxide. So notice how I wrote 2 acetyl-CoAs. 2 acetyl-CoAs would make 2 ATP, 6, and 8. But if I'm talking about 1 acetyl-CoA, just make sure to divide everything by a half, right? So 1 acetyl-CoA can only make 3 NADH, not 6, right? And last step. So you guys can see I wrote the NADH and FADH in different colors. So what's going to happen to this blue and purple, right? we're going to take these electron carriers, which we made in glycolysis, pyruvate oxidation, and citric acid cycle, and we're going to finally use them in oxidative phosphorylation to make energy. So I got this from Dr. Joseph's slide. It's pretty much showing you that in the inner membrane of the mitochondria, we have these proteins, which you can see in blue, that are pretty much pumping protons. And so what happens is we're taking our electron carriers, which is our NADH and FADH2 that we made in different steps, right? We're taking them and we're taking their electrons, so we're oxidizing these carriers, right? And we're pumping protons across the membrane. So now our inner membrane space is going to have a high concentration of H+, so it's going to have a low pH in the inner membrane space. And because of this chemical gradient that we've created, these protons are going to spontaneously flow down back into the matrix. And so because of this, we're going to create energy to drive the synthesis of ATP. So since these protons are moving down their gradient, it's going to be a spontaneous reaction, which we can couple to a non-spontaneous reaction, which is ATP synthesis, right? So if we look at the reactant side, this is all generated from glycolysis, pyruvate oxidation, and citric acid cycle, right? So we're taking our electron carriers, which is 10 NADH, 2 FADH2s, and we're using this to make ATP. So these electron carriers, the 12 electron carriers, will generate 28 ATP through oxidative phosphorylation, right? So if we go back to our chart, we took the 10 NADH and the 2 FADH2s. Again, if you add up all the blues and purples from the other steps, you would get that number. And we made 28 ATPs. And this is all occurring in the mitochondria. But remember, 28 isn't the total. So we have to take the 28 that we made through oxidative phosphorylation. We go to the next slide. Along with the Krebs cycle ATP and the glycolysis ATP, giving us a total of 32. So I definitely recommend taking a picture of this chart and adding more to it, or making your own and kind of filling in different information if you guys need to, because that will be really, really helpful. Good luck.